Okay. Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. In this video lecture, we are going to talk about aminoglycoside. Now, this is another video lecture which are regarded with the antibiotic. And in this video, we are actually to talk about the aminoglycoside. So let me write here. Aminoglycosides. Now, these drugs are actually the protein synthesis inhibitor drugs. These are the protein synthesis inhibitor drugs. Because these aminoglycosides are usually used to stop the protein synthesis. These aminoglycosides are actually the 70S inhibitor. These are the 70S inhibitor. If you remember, in the previous videos lecture, we talked about the 50S inhibitor, we talked about the 30S inhibitor. Now this is another video, this is about the 70S inhibitor. So this aminoglycoside is actually 70S inhibitor. It means that these aminoglycoside will usually responsible to stop or to prevent the assembly of 70S ribosomes, means there is a 30 and 50S. So due to the giving of these aminoglycosides, so 70S ribosomes, or we can say 70S or complete assembly of the ribosomes will be not occur. So complete ribosomes will be not formed whenever we are giving these antibiotics. Now the drugs which are used in these antibiotics, these drugs are usually streptomycin, kanamycin, gentamicin, neomycin, tobromycin, and some other drugs as well. So whenever this mycin comes at the end, then these drugs are usually mentioned in aminoglycoside. So whenever you want to memorize, the drugs, so you should have to remember this mycin at the end. So whenever these mycin at the end are come, so these drugs are actually the aminoglycoside. Now, how this aminoglycoside will be look like? So I'm not drawing the exact and the correct one structure, but I'm drawing the simple or the actual, that how the aminoglycoside will be look like. Aminoglycoside will be look like somehow in this form. If you see in Google, so it will be look like in this form. So this is not the exact structure, but it will be look like in this form. So this is the aminoglycoside, means it contains 5 carbon ring. So these are the aminoglycoside will be look like in this form. Not correct, but actually in this form. Now let's talk about the mechanism of action of this aminoglycoside. Now before going to explain the mechanism of action of these drugs, let me tell you some important thing that I told you, that whenever you want to memorize or whenever you want to remember the mechanism of action of these drugs, first of all you need to understand that how a protein is synthesized. I already explained that how a protein is synthesized. If you missed that video, so go back to my channel and watch the video that how actually protein is synthesized. So once you get the idea about the protein synthesis, then what actually happened in mechanism of action? In mechanism of action, let's suppose if this is the mRNA, means messenger RNA, mRNA. So this will be the 30S ribosome. And this 30S ribosome, as I told you, it contains 3 sites. A site, P site, and E site. Whenever the 30th ribosome and this messenger RNA are bound together, then we actually call that pre-initiation complex. PIC means pre-initiation complex, when the 30th and this messenger RNA are bound together. Then what actually happened, I also told you that this A site will contain a release vector. This black one represents the release vector. What actually happened, when the transfer RNA, let's suppose this transfer RNA, let me draw with the with the green marker. This is, let's suppose, a transfer RNA. They have the anticodon. When they bind to this P site, so this release vector will released out. So when the release vector is released out, due to this way, the 50th subunit are come and bind to that area. This release vector is actually responsible to prevent the 50th subunit to bind that area. So this release vector is not allowing the 50th to bind to that area. When that transfer RNA bind to that area, then the release vector will release out. After that, then a 50th subunit will bind. And then again 50th subunit have again these three sites, A, P, and E. So this is the 50th. So when 30th, 50th plus messenger RNA, these three are combined together, then we call that translation initiation complex. We call that translation initiation complex. Now what actually happened in mode of action of this aminoglycoside? This aminoglycoside, let's suppose this one drugs, this one drugs will come and bind to this site, to this A site with the release vector. They bind to this A site with the release vector. So let's suppose, let me draw with a red marker. So let's suppose these are the drugs. They bind to that area. These are the aminoglycoside, let's suppose. Not the exact, but this is, for example, this is the drug. So whenever they bind to this release vector, then the release vector will be not released out. So when the release vector is not released out, so how the 50th subunit are bound? So when the 50th subunit is not bound, then other transfer RNA is not bound to this A site. Or we can say the protein starting point will be halted. So this is the first mode of action of this aminoglycoside. So we can say this is the first one. This is actually initiation. Or we can say at the beginning of protein synthesis, at the beginning of protein synthesis. So at the beginning of the protein synthesis, or in case in initiation stage, these drugs will perform their mechanism of action. Now this is the first mechanism of action. So whenever they bind to that area, so what actually happen? Complete 70th ribosome will be not formed. Because 50th, although 30th is present, but the 50th subunit will not bind. So when the 50th subunit is not bind, so the complete 70th ribosomes will be stopped. Or so that's why we can say that it is the 70th inhibitor, because complete 70th ribosome is not formed. So this is the first mode of action. The second mode of action of this drugs is that they can perform their mode of action at the end of the protein. When the protein is synthesized, then at the end, these drugs will perform their mechanism of action. So this is the second mode of action, means at the end of protein synthesis. Or we can say termination. So at termination, in termination stage, this aminoglycoside will show their mode of action. How? Let's suppose again in this case, let's suppose, let me draw with a blue with a green marker. Let's suppose this is again a messenger RNA, and this is the 50th subunit of the ribosome. This is the 30th subunit of the ribosome. These are the sites which are present inside the ribosomes. And this is the transfer RNA which is bind to that area. And these are the polypeptide chain, or we can say these are the amino acid. A long polypeptide chain will be formed. And then what actually happened at that area of the messenger RNA, at that area of the messenger RNA, there are some stop codon are present. So these are what? These are actually the stop codon. The stop codon. And these stop codon are actually UAA, UGA, and UAG. Fine. So these are the stop codon. So whenever the stop codon are present, or whenever the ribosomes are scanning and reach to the stop codon, then the release vector, then another release vector. So let's suppose this is a release vector. Release vector, in this case, let me draw this in this form. This release vector will have two domains. This one is actually responsible for the hydrolysis. And these one are the actually anticodon. Now what actually happened? They will come and sit to this A site. And this hydrolytic site will bind to this long polypeptide chain. Now, due to the binding of this hydrolytic site with this polypeptide chain, then they will cleaved. So whenever this long polypeptide chain are cleaved, so they will release out and they will arrange their shape secondary or tertiary, we can say, or because you better know that protein have three main structure, sometimes four: primary, secondary, and tertiary, or so primary, secondary, and tertiary. So they will arrange their structure. So these are actually polypeptide chain, or we can say these are the protein polypeptide chain. So these are the polypeptide chain. Now then these polypeptide chain are actually responsible for the protein. Now what are the mechanism of action of this aminoglycoside during termination? Let's suppose at that area there is no release vector. Let's suppose at that area there is no release vector. First of all, the drugs. Now let me. This is the exact structure, but what actually happened? Let's suppose this is the drugs. The drugs will bind and occupy that area. This is the drugs, or we can say this is the aminoglycoside. These drugs will bind. This aminoglycoside will bind to this A site, and they will not, or we can say they will not allow this release vector to bind to that area. Why? Because here already the drugs are bound. So when the drugs are bound to this A site, so there will be no space for the release vector. So whenever the release vector are not bound to this A site, so how it is possible? Means, so how this polypeptide chain will be cleaved? How this polypeptide chain will be cleaved and released out from this ribosomes and responsible for the functioning. So this is the second way of the. So this is the second mode of action of these drugs. Now this is the second mode of action. So we can say, so whenever these release vector are not bind to that area, so we can say that actually this drugs is responsible to bind to this release vector, to block the release vector and to prevent the release vector to release out. This is the first. And in this case, they will bind to that area and not allow the release vector to bind with this area. So these are the two mechanisms of action of these drugs. So both are opposite to each other. So that's all about the mechanism of action of these drugs. So we can say that these drugs is actually bacteriostatic in nature. Bacteriostatic in nature. So these drugs are bacteriostatic in nature. Why? Because they can stop the growth of bacteria. Now, what are the mode of, not mode of action, what are the antibacterial spectrum of these drugs? Antibacterial spectrum, or we can say mood, or we can say that broad spectrum as well. Now, what are the antibacterial spectrum of these drugs? These drugs are activated and show good results. Against Pseudomonas aeruginosa, E. coli, Proteus, Klebsiella, Shigella. So against these bacteria, the Pseudomonas aeruginosa spelling is not the right one spelling. It is actually wrong, but I don't know that what are the exact spelling. Then you can check the exact spelling. So these are the bacteria. These are the bacteria which are affected by giving, whenever we are giving these drugs. So there are some other bacteria as well, but these are the exact bacteria. So these are the antibacterial spectrum of the this aminoglycoside. Inshallah, in the next video we will talk about the resistance which are activated against these drugs. So please stay tuned and continue to watch. Thank you so much.